Dear students, in this session we are going to discuss the second stage of aerobic respiration. In our previous session we have learnt about glycolysis, in which glucose 6-carbon molecule splits into 2 molecules of pyruvic acid by involving a 10 series of reaction which takes place in cytoplasm. The second stage is Krebs cycle. The Krebs cycle takes place in mitochondria, matrix of mitochondria. So in Krebs cycle, pyruvic acid undergoes decarboxylation reaction, means removal of carbon dioxide molecules, and oxidation reactions, removal of hydrogen ions. The pyruvic acid undergoes complete oxidation and decarboxylation reactions in Krebs cycle. So these reactions, the oxidation and decarboxylation of pyruvic acid, these reactions, this series of reactions are described by Hans A. Krebs. That's why it is also called, it is also popularly known as Krebs cycle. So Krebs cycle has one more name due to, in Krebs cycle, the first formed compound is citric acid. The citric acid contains 3 COOH functional groups. 3 COOH functional groups. These are carboxylic acid groups. So due to presence of 3 carboxylic acid groups in citric acid, the Krebs cycle is also known as tricarboxylic acid cycle. Tricarboxylic acid cycle. Okay, let us discuss the series of reactions, the metabolic pathway of Krebs cycle, step by step manner. The first reaction is known as condensation. Condensation means here 4 carbon oxaloacetic acid combines with 2 carbon acetyl CoA. Already I told you the pyruvic acid enters into Krebs cycle in the form of acetyl CoA. The pyruvic acid undergoes decarboxylation and oxidation reaction. As a result, pyruvic acid converts into acetyl CoA. The acetyl CoA enters into matrix of mitochondria. Okay, the first reaction is the condensation, in which 4 carbon oxaloacetic acid combines with 2 carbon acetyl CoA in the presence of citric synthetase enzyme. So it results in the formation of 6 carbon compound, that is citric acid. Citric acid. I told you citric acid contains 3 COOH functional groups. That's why it is called tricarboxylic acid cycle. Next, the citric acid undergoes dehydration reaction. Dehydration means removal of water molecule. So this reaction catalyzed by aconitase. As a result, citric acid converts into cis-aconitic acid. The next reaction is hydration, means addition of water molecule. Here one water molecule is added in the presence of same aconitase enzyme. As a result, cis-aconitic acid converts into isocitric acid. Isocitric acid. So the isocitric acid undergoes oxidation reaction. The first oxidation reaction is the isocitric acid undergoes oxidation in the presence of isocitric dehydrogenase enzyme. So I already told you respiration is the oxidative reaction. The all oxidation reactions of respiration are removal of hydrogens, only removal of hydrogens. Here the isocitric acid lost hydrogens, so these released hydrogens received by coenzyme NAD to form NADH. So this reaction catalyzed by isocitric dehydrogenase. As a result, isocitric acid converts into oxalosuccinic acid. Next, oxalosuccinic acid undergoes decarboxylation. Here, oxalosuccinic acid, also 6 carbon compound, it undergoes decarboxylation in the presence of oxalosuccinic decarboxylase. As a result, oxalosuccinic acid converts into 5 carbon compound, that is alpha ketoglutaric acid. because oxalosuccinic acid loses one carbon in the form of carbon dioxide. Next, oxidation 2. రెండవ ఆక్సీకరణ చర్య అంటాం. ఆక్సిడేషన్ 2, the alpha ketoglutarate acid undergoes decarboxylation, means removal of carbon dioxide molecule, and oxidation, the removal of hydrogens. So alpha ketoglutarate acid undergoes decarboxylation and oxidation. Here one more interesting coenzyme A is also added in this reaction. As a result, alpha ketoglutarate acid converts into succinyl CoA. Here the released hydrogens received by NAD to form NADH. The next reaction is cleavage. This means split. Here one interesting thing, in respiration is an oxidative reaction. All oxidative reactions are involves the removal of hydrogens, the received by NAD/FAD to form the reduced FADH to NADH2. But one interesting thing is, in respiration, in aerobic respiration, when substrate split, some amount of is released, these released energy captured, utilized to form ATP molecules. So this process called as substrate level phosphorylation. చూడండి, ఇక్కడ సక్సినైల్ కోఏ విచ్ఛిన్నం చెందినప్పుడు, స్ప్లిట్ అయినప్పుడు విడుదలయ్యే శక్తితో కూడా మనకు ఏటీపీలు డైరెక్ట్గా ఏర్పడతాయి. సో దిస్ ఫినామినా ఈజ్ ఆల్సో నోన్ యాజ్ substrate level phosphorylation. Succinyl CoA undergoes cleavage reaction in the presence of succinic thiokinase. Succinic thiokinase, the enzyme that catalyzes the cleavage of succinyl CoA to form, as a result, it forms succinic acid and coenzyme is separated. So the energy released during split of succinyl CoA utilized to form ATP molecule. This phenomenon is also known as substrate level phosphorylation. The next, succinic acid undergoes oxidation reaction. Oxidation reaction. The succinic acid undergoes oxidation reaction. In this oxidation, the released hydrogens received by FAD to form FADH2. As a result, succinic acid forms fumaric acid. Next, fumaric acid undergoes hydration, means addition of water molecule. In the presence of fumarase enzyme, fumaric acid converts into malic acid. The last reaction in Krebs cycle, malic acid undergoes oxidation. The oxidation 4 reaction, the malic acid undergoes oxidation in the presence of malic dehydrogenase enzyme. So in the oxidation, the released hydrogens received by NAD to form NADH. So as a result, malic acid converts into again oxaloacetic acid. It is the series summary of reactions which takes place in Krebs cycle. Okay, the final result of Krebs cycle is 1 pyruvic acid undergoes 4 oxidative reactions. In 3 oxidative reactions, the hydrogen acceptor is NAD. So as a result, for oxidation of 2 molecules of pyruvic acid, 6 NADH are generated. Again, for 2 molecules of pyruvic acid, during conversion of succinic acid into fumarate, in this oxidation reaction, the hydrogen acceptor is FAD. So that 2 FADH are also generated. So 2 ATP molecules are formed, generated in the process of substrate level phosphorylation, where succinyl CoA splits to form succinic and coenzyme A. So 4 CO2 molecules are released. So the reduced coenzymes like NADH and FADH involved entered the third and final stage of aerobic respiration, that is electron transport. Electron transport, where these reduced NADH or FADH undergoes oxidation. They finally form the ATP molecule. They generate ATP molecule in the light of chemiosmotic theory proposed by Mitchell, which states that the proton gradient act as a driving force of for ATP synthesis. So it is the comprehensive details of Krebs cycle. Thank you.